Hi and welcome back. This is the second episode in the series of videos discussing cell injury. In the previous video, that is part 1 or episode 1, we introduced ourselves to pathology and discussed what is cell injury, the various adaptation disorders, the causes and the mechanism or pathogenesis of cell injury. If you haven't watched that, do watch it now. The link is available in the description and is also now available on the screen. In this part, that is part 2 or episode 2, we will be discussing the morphology of both reversible and irreversible type of cell injury. We will be covering the morphology of reversible cell injury or the features of reversible cell injury, and we will also be covering the features of necrosis, that is a type of irreversible cell injury. Okay, let's jump into the subject. The reversible cell injury includes hydropic change, fatty change, hyaline or mucoid change. First, we will discuss the hydropic change. Hydropic change is also known as cloudy swelling or vacuolar degeneration. It is caused due to intracellular accumulation of sodium and water. In our earlier video, while discussing the pathogenesis of cell injury, we saw that due to depletion of ATP, there is defective functioning of sodium potassium pump. This defective functioning of sodium potassium pump leads to escape of potassium from the cell and influx of sodium into the cell. I also told you wherever sodium is there, water follows it. Influx of sodium is followed by influx of water. Sodium carries water with it wherever it goes. Okay. So along with sodium, there is also intracellular accumulation of water. And this leads to hydropic change or cellular swelling. Here, the cell is enlarged, swollen, with clear vacuoles in it. There may be cytoplasmic blebs. You can see the swellings here. These are called blebs. There can be cytoplasmic blebs. Coming to the next type of reversible cell injury, that is fatty change. Fatty change or steatosis refers to intracellular accumulation of neutral fat within the parenchymal cells. That is intracellular accumulation of lipid, that is fat. It mostly occurs in liver, and in that case it is called fatty liver, and fatty liver occurs in conditions like the conditions with excess fat like obesity, diabetes mellitus, or the conditions of liver cell damage. The most common one is alcoholic liver disease. As you know, free fatty acid is esterified to triglycerides and then converted to lipoproteins by fusing it with a lipid acceptor protein. So the free fatty acid is esterified to triglycerides and converted to lipoproteins and then released into circulation from liver cells as plasma lipoproteins. The very famous LDL, VLDL, which you might have already known. In fatty liver, there is intracellular accumulation of these triglycerides due to defect in this process. The defect can be either increased entry of free fatty acids into the liver or increased conversion of free fatty acids into triglycerides or decreased synthesis of lipid acceptor protein or decreased availability of lipid acceptor protein to convert it into lipoproteins or blocked excretion of lipoproteins, which can lead to accumulation of triglycerides in these liver cells. In this case of fatty liver, the feature is the liver cell or hepatocyte is filled with lipid vacuoles or microvesicles of fat. Initially, these are small and present around the nucleus. Later on, the vacuoles become larger, pushing the nucleus to the periphery, occupying the entire cell. In usual HND sections, these appear as clear spaces because in normal HND staining, due to the organic solvents used, this fat is dissolved. So it appears as clear spaces, but it can be seen or demonstrated by special staining techniques for fat like Sudan dyes. Now coming to morphology of irreversible cell injury, we have to discuss the morphology of two types, necrosis and apoptosis. We will discuss the morphology of necrosis in this video, and apoptosis we will be discussing in our next video. Necrosis refers to the spectrum of morphologic changes following cell death. That is, it is the death and following degradation of cell. The degradation which follows the cell death can be caused due to the hydrolytic enzymes liberated from the dead cell itself or from other cells, that is the inflammatory cells which reach the place. Based on that, it can be called either autolysis or heterolysis. Autolysis, where the degradation occurs from the hydrolytic enzymes liberated from the dead cell itself, and therefore there is no inflammatory reaction. And heterolysis, the degradation occurs due to the hydrolytic enzymes released from the inflammatory cells, and hence it is invariably accompanied by an inflammatory reaction. The two major events occurring in necrosis are the denaturation of cellular proteins and enzymatic digestion of cell. There are 5 types of necrosis: coagulative, liquefactive, caseous, fat, and fibrinoid. Coming to coagulative necrosis, the feature of coagulative necrosis is that the basic structure of cell is retained. That is, the cell type can still be recognized, whether it is a columnar cell or cuboidal cell. The cell type can still be recognized because the basic outline of the cell is retained. Only the cytoplasmic and nuclear details are lost. The hallmark is the normal cell, as you can see here, is converted to its tombstone or outline, as you can see in this diagram. The normal cell is converted to its outline or tombstone. So the basic structure is retained, only the cytoplasmic and nuclear details are lost. The cell type is recognizable, that is coagulative necrosis. Whereas in liquefactive, otherwise called coagulative necrosis, the entire thing is converted into a liquid viscous mass. This occurs due to the tissue degradation by hydrolytic enzymes. The most common example is infarct brain. The affected area is soft with liquefied center containing the necrotic debris. Coming to caseous necrosis, this one combines the features of both coagulative and liquefactive necrosis. This is often found in tuberculous infections. The foci of necrosis resembles dry cheese. That's why it is called caseous. The focus contains structureless eosinophilic material having scattered cheesy granular debris of disintegrated nuclei. And of course, it is surrounded by the granulomatous inflammatory reaction. Coming to fat necrosis, this occurs in fat-rich anatomic locations like the breast, or in the case of acute pancreatitis. Here, the feature is a release of lipases. There is hydrolysis and a rupture of adipocytes releasing neutral fat, which changes into glycerol and free fatty acid. The leaked out free fatty acid sometimes complexes with calcium to form calcium salts. The necrosed fat cells gives a cloudy appearance and is surrounded by inflammatory reaction. The fibrinoid necrosis is characterized by deposition of fibrin-like material in blood vessels. It is encountered in examples of immune-mediated tissue injury or immunologic tissue injury. Here, there is immune-mediated protein denaturation. With this, we will end this episode. In the next episode, we will be discussing the morphology and mechanism of apoptosis, followed by changes after cell death like gangrene and calcification. As usual, PDF notes of this topic is available for download in the description in the link. For further guidance or live interactive sessions, you can mail me. For more videos, keep the channel subscribed. Until we meet in another video, thanks for watching. Bye.